RNA interference versus CRISPR in an experiment to eliminate or reduce expression of gene A, and which one we choose and why. They are both approaches for gene silencing. Rnai, or interference, is double-stranded RNA initiates the silencing. It's then cut and diced into small fragments by an enzyme called dicer. Those small fragments then bind to a protein that remove one strand of RNA, allowing the other strand to bind to the target messenger RNA. This protein can either then cleave, which is cut, or recruit accessory factors to help regulate the gene expression. It's naturally found in eukaryotic cells and used as a gene regulator. It's considered a knockdown, meaning that the expression of the gene is reduced or silenced, not completely destroyed. The silencing of the gene is temporary. CRISPR uses an enzyme that is able to cut apart DNA. A guide RNA is made, and the enzyme is able to find this precise match. It unzips the DNA, pairs it, and then it is cut both strands. Naturally, the DNA will try to repair itself, and this may include turning the gene off, repairing the gene with better or worse features, or we can actually insert new DNA that we desire to replace that cut DNA. Naturally, it's found in prokaryotic cells, and it's used as a defense mechanism. It is considered a knockout, which means it completely destroys or removes the DNA. The silencing is permanent. Which method would I choose to learn more about gene A with silencing in my favorite eukaryotic cell? I would choose Rnai. And the reason that I would choose that is because you can temporarily silence the gene, meaning that we can turn that gene off, observe what the expression, what happens with the expression, and then we can turn that gene back on. And then when we turn it back on, we can also observe what happens and confirm or verify that it is that gene that is related to that expression. With CRISPR, it completely deletes the DNA, so we're not able to turn it back on again. So once we do that, that's it, that's done. Now, if we wanted to add our own desired DNA template to it, then we'd want to use CRISPR because that would allow us to be able to cut that DNA and then insert the new DNA that we would want. And that's why I would choose Rnai.